right, we're settling back in for the next section. So it's my great pleasure to introduce Dr. David Novo. He's certainly someone who doesn't need any introduction here, but we'll certainly go ahead and indulge him. David received his PhD in biophysics from UCLA. His research concentrated on developing novel optical and mathematical techniques for measurements and analysis of localized fluorescent calcium transients in muscle cells. Throughout his career, David has made many invaluable contributions to the field of flow cytometry, including the development of methodology of bacterial analysis by flow cytometry, oversight of the FCS file standard, and most notably as the founder and creator of FCS Express data analysis software, which I'm sure many of you in the room have used. David has published extensively on pioneering data analysis strategies, exemplified by his most recent publication titled Reducing Spreading, Removing the Impact of Irrelevant Dyes Improves Unmixed Flow Cytometry Data, which introduces a novel unmixing method called True OLS, which utilizes a priori biological knowledge to unmix each event with the only dyes present on the event to decrease the variance of unmixed abundances, both statistically and visually, in simple and complex staining panels. So it is my great pleasure to introduce, please give a warm welcome to David.
Thank you. Thank you, everyone, and thanks for coming to my talk today, and of course, thanks all the organizers for inviting me. It's always nice to come back to New York and a lot of familiar, friendly faces, especially now that I don't have to deal with their software bugs and they don't blame me for it. They're much friendlier than they used to be. So what I'm going to try to do today, so I do apologize in advance, is the very first time I've given this particular talk. I rewrote it for MetroFlow, so I'm sure I'm going to notice 50 mistakes during the talk, and if any of you do notice any as well, please let me know, because I'll fix it and try to get it perfect. But what I'm going to try to do today is kind of give a background to some of the causes of so-called spreading in flow cytometry data. Try and give an introduction to some of the mathematics that goes into it in a way that I hope is digestible to people, and kind of follow up with, now that there's, now that you guys have some background, with some of the new work that recently published to try and help mitigate some of the problems. So with that being said, okay, so the first thing to do is, before we start, is to define what is spread. You know, the word is kind of thrown around a lot, and when you ask people what it is, it's just kind of defined as the spreading of your data. It's a bit of a circular definition. So what I'm going to do today is define spread as the variance in your populations, the non-biological variance in your population. So the variance that you see in your histograms or in your dot plots that is in excess of the true biological variance of whatever sample you're trying to measure. Okay, so that's what spreading is going to be. And what is, you know, this, I'm going to go through this part very quickly because I'm sure people are very familiar, but for those that are not, what is the problem? The problem is that photons from any particular dye are detected in multiple detectors in your system. So for example, from the FITC dye, you're going to get 500 photons detected in this first detector, but 150 photons in the second detector. I'm using the term here photons very loosely. It's more complicated than that, but for the sake of the argument, we'll just say that that's what's happening. And then from the yellow dye, we're going to get a signal of 5 photons show up in that first detector again, 400 in the second detector, and 5 photons show up in the third. And then for our last Per-CP dye, we get 500 photons showing up in this third detector and 100 in this fourth detector. And of course, this is all fine and good, except for the fact that we actually don't see that. We only see this bottom measurement here, 505, 50, 505, and 100 in each detector. So we get what's called an observation, or the detected signal, and we know that the detected signal contains a mixture of photons from all of the different dyes. We just don't know what they are, right? This is not new to anybody. And so what is the compensation mindset? So this is historically sort of the way people understood the problem, or is that there's some sort of error that happened that you need to compensate for it, right? Even in the very terminology that people are used to doing in flow cytometry, we're doing compensation, we're compensating for some sort of problem that happened, right? And the idea was, if you noticed, I didn't call them the FITC detector or the PE detector. I called it detector 1, detector 2, detector 3, detector 4. But historically, this has been known. There was generally in your instrument a FITC detector. There was a PE detector. There was a PerCP detector. And that's the way that they were referred to, with the idea that that is the detector that those dyes were supposed to be, quote unquote, go into. If you could magically like shove them into a detector, that's the detector they were supposed to be. And so there was a primary channel for each detector that the fluorescence was supposed to be in. And the problem is the fluorescence was pesky and it went into detectors where they weren't supposed to be, quote unquote, right? And so we needed to correct that by correct the detected values that we detected in our primary detector for the signal that spilled over into that detector from all of the other detectors, right? So somehow each detector was somehow polluted by signal that spilled over from other detectors, and we were going to compensate for that by doing something, right? And so again, here's the components and here's the dye, right? And the idea is somehow that we were going to correct the FITC detector by taking what we were doing, the thought process or the understanding of what we were doing, is we were taking this FITC detector and we were taking the signal that we got and we were subtracting out the amount of signal that we got from the other detectors that spilled in to come back to the true amount of signal that we should have gotten if FITC was there by itself, right? So the FITC detector, we got 505, but 5 of those were really from the PE, so we're somehow magically subtracting them out. We get back to 500, which is what we should have gotten, quote unquote. And for PE, we got 550, but we're subtracting out those 150 pesky photons to get back to the 400 that we should have gotten if that signal was a pure PE signal, and all the way along. And so I feel like this is the understanding that the conceptual framework that people had regarding what we were doing when we were doing compensation. And again, this was pervasive throughout the field, okay? So if we look at one of the classic papers, which has most of the useful information that's needed to do compensation, and was an amazing paper that was the guide. Even this paper had this understanding was so prevalent that even in this paper, a lot of this kind of concepts were in there, right? If we look at this, however, some FITC fluorescence appears in the PE detector, right? So referring to a detector by the fluorochrome that is supposed to be there because of the emission overlap. The signal is termed spillover because it spills from the PE detector to from the FITC detector to the PE detector, right? And from the same paper, this is termed compensation, correcting the PE signal for the amount of FITC fluorescence in the PE band. So here he's a little more subtle actually about not actually calling it a PE detector. And then again from the paper, note that while the spillover from FITC to PE Sci5 is 4%, if you look in the actual table that he's referring to, he is actually very clear that he makes a separation between the fluorochrome and the detector. They're not the same thing. He labels them in 2 different ways. One is the red detector and the green detector, and here are the dyes. But it was the thought process is so pervasive that when writing the text, neither Mario nor the reviewers noticed that he actually basically referred to this as the 4%. is actually going to the red detector, but it was referred to basically as the PE-Sci5 detector. So the idea is very pervasive that you're correcting the detectors and fixing the detectors because the detectors belong to a certain dye, okay? And it's actually never been that way. That is not a useful or helpful way to think about the problem, and it's not actually correct what we were actually doing when you do compensation. Okay, and I'm going to walk through a small example here. I feel like this laser pointer is not so great. So what we're going to do is I made a very simple mixing matrix over here. This is a compensation mixing matrix. You've got 4 colors and 4 dyes, one along the diagonal. And these are what our 4 little simulated dyes look like. Okay, each dye, I designed this so every dye has a peak in a particular detector. So every dye, this would be the FITSI dye because it's the most FITSI if we want to think about it the way that people were. This is the PE, this is the PerCP, here's some other dye. And here is a not unrealistic compensation matrix that I generated for this. Okay, and if you actually look at it, if you take these 4 made up abundances of each dye, this is how much dye there actually was, and you multiply it by this mixing matrix, you get the observation that you would have gotten from these 4 dyes given this mixing matrix. And the observation looks like this. This is the value you would have gotten in every detector from our simulated experiment over here. Okay? And in fact, if you take this observation and you multiply it by the inverse of the mixing matrix, if you multiply it by what was termed the compensation matrix, you actually perfectly recover the abundances of the 4 dyes that went into it, right? So this is sort of what, this is what happens when we're doing compensation, right? You have an observation from the flow cytometer. You don't know what went into this observation. This is just what you got out of the detectors. You multiply it by your compensation matrix and you get back some abundances, and lo and behold, these are the actual abundances that I put into the systems showing that the compensation actually worked, right? But let's see what is actually going on. And what's actually going on is if you look at, this is the compensation matrix from the previous slide, I just copied it over here. And if you actually look at the math, in order to calculate the amount of dye 1, you are taking about 1.9 times the value in detector 1. And in fact, you are subtracting out some of the signal from detector 2. So whatever was in detector 2, you are subtracting out some of that signal. Dye 2. So whatever value you got from for the detector 2, which is from the second dye, you are subtracting it out. But then you are adding back the signal that you got from some signal from detector 3, and you're actually adding some signal from detector 4. You're not simply subtracting out spillover values saying, oh, well, in detector 1, I got this, but then some of the dyes spilled over into other detectors, so I'm subtracting it out. You're actually adding things back in from other detectors, okay, in order to recover what was your primary detector. Again, the idea being that if this was the primary detector and this was the secondary, if this was where my corrected detector 1 and this was my real detector 1, I should be subtracting out the spillover from the other dyes. And in fact, you actually never are. If you look at this one here, for example, in order to calculate dye 3, you are subtracting out some surrogate from detector 1, you are subtracting out some surrogate from detector 4, but you're actually adding back in some of the signal that was in detector 2 in order to create dye 3. But if detector 2 was primarily from your second dye, you should be subtracting out this kind of dye over here in order to recover what should have been here. Okay, so you are never, ever, we never have been simply taking the value in a certain detector and then subtracting out signal from other detectors in order to recover the true signal, in order to recover the true FITC detector value that should have been there if the other dyes had not been there spilling over and polluting my FITC detector. Okay? So the math is much more complicated. So what are we actually doing? So what we're actually doing, and Mario might have said it earlier, this is the first paper. So Mario changed his terminology to be much clearer as time went on. And this is what we're doing, okay? So the process of compensation is a transformation of measurement space into fluorescence space. We are literally transforming the values we get from the detector, the measurement values, into what we're going to call today an abundance value, which is the amount of fluorescence from that particular dye that's on the cell. Measurements from any single detector compromise contributions from multiple fluorescence because of spectral spillover. With standard linear algebra, these are transformed into values to estimate the pure individual fluorescences, okay? So we are literally doing a transformation from detector space, which are the measurement values that we get, to a fluorescent space, which is the amount of fluorescence on every cell. There is no correcting detectors going on. We are going from one completely separate concept, which is a measured value, to another completely separate concept, which is an amount of fluorescence. Okay? And spectral makes this obvious, right? Once we get into spectral flow, it's obvious that there's no primary detector. Which is the primary detector here for any particular dye? The question doesn't even make sense, right? So we can't actually be correcting the primary detector for the amount of dye that's spilled over into the secondary detectors. It makes it obvious here that what we are doing is transforming this, whatever, 30-something detector system that I have. We're taking 30 measurement values and transforming them into 3 dye abundance values in this particular case. And again, so this is, I said, which one should be secondary? It's obviously a silly question, right? And so I've talked to people who've said, yes, okay, this makes perfect sense. Here we definitely are taking 30 detectors and transforming them into 3, so it's obviously that we're not correcting any particular detector. But compensation and unmixing are not exactly the same, and in some magical way, that while the operations look similar, because compensation is a square matrix, you have the same number of detectors as you have dyes, and unmixing or spectral is a rectangular matrix, you have more detectors than you have dyes, then somehow that gives these two processes slightly different interpretations, and you should think of it as, in compensation it was correct to think about it the way that people were. This is quite simply incorrect, okay? Compensation and unmixing are literally exactly the same thing. Okay, it just, the size of the matrix makes the math happen slightly differently and simplifies the problem a little bit in the compensation sense. It is exactly the same thing, and in both cases, you are transforming your data from a measured value into from a measurement detector value that came out of the detector into a abundance value that is nominally something proportional to the amount of fluorochrome on your dye. Okay, so if that is the only thing you remember from this, and it's funny because people who are new to flow cytometry are like, okay, and people who've been doing this for a long time, it takes a while to wrap your brain around it because the way of thinking about it before is just so pervasive in the field. And it's not, and it never was 100% correct, and for sure now it's not useful any longer for reasons that I'll get to as we move along, okay? So you are going from one space, which is measured in channel values or detector values, whatever you want to call it, arbitrary units, into another space, which is measured, the units are debatable, based on what kind of corrections you perform. I just like to think about it that this is photons, fluorescent photons from the dye, and this is an electrical signal that's coming out of your flow cytometer, and you're converting from one thing to another. Okay, so how do we do this conversion, right? We're going from raw observations into dye abundances. And we do that by plugging our data through a mathematical model. This is another thing that I do want to stress during this talk, is that compensation was very much thought of by many people, if not most people, as a somehow simple correction of the data. I am just correcting the data like I am making fine-tuning adjustment on a scale, like I press tare and it goes back to 0. It's just a very simple correction. I'm just subtracting out some things that shouldn't be there, and I'm getting back to the answer that I want. And that's not true either, okay? And it never has been true. Okay, and what I'm going to stress for the next few slides is that we are actually applying a mathematical model to these raw observations in order to recover these dye abundances that are here. Okay, and flow cytometry is no stranger to mathematical modeling. Okay, we have been doing mathematical modeling for a long time. People have mathematically modeled, taken a histogram, right, and mathematically modeled it, that out of this 128 or 256 channel histogram, they get out 3 things: %G1, %S, and %G2, right? And out of a proliferation histogram, you have a 256 channel histogram or 1024 channel histogram, and out of this you transform it, you apply a model, and you transform it into a completely other space, which is the percentage of cells that are in every generation that divided, right? The original data has nothing to do in terms of units or concept with the original data that you're generating, histogram channels, is completely transformed into a completely separate entity, which is the %S, %G2, whatever, right? And people, I think, conceptually saw this process as somehow different than compensation. Compensation is like a mechanistic thing where you just correct the data. It's not a model, it's just some sort of tweaking some dials. And then this is actually, of course, mathematical modeling, right? And I'm here to say that they're all exactly the same. Unmixing is exactly as much mathematical modeling as DNA cell cycle analysis or proliferation analysis, okay? And once you are able to accept that they're exactly the same thing, then it kind of opens the door to applying all of the techniques and understanding compensation in terms of many of the things that people know about mathematical modeling. The first one being that mathematical modeling is only as good as the assumptions that underlie your mathematical model, right? You'll always get a result from a mathematical model. When you plug a model into your data and you run it through an algorithm, you'll always get an answer. And it's pretty, I think, universally understood that if that model that you plugged it into reflects the underlying process that generated the data, then the results that you get from the model will be useful for you. And if the model does not match the underlying process, then the results might not be so useful, right? And for people who do cell cycle analysis, they're very used to this, right? There's like 6 variations of a cell cycle model, and you can choose whether to include aneuploid cells or non-aneuploid cells or sub-G1 or non-sub-G1. And if your data had that stuff and you don't include it, then your model will not come out correct. Your S-phase estimations will be wrong. And if your model didn't have the stuff and you did include it, then your S-phase estimations will be wrong as well. And so you've got to match the mathematical model to the data that you got for cell cycle. And it's the exact same thing for unmixing / compensation, which I will use just unmixing to mean both, okay? And so what is the mathematical model that we are applying in the unmixing algorithm? And the mathematical model is that I've got an observation from a single cell, and I am assuming that that is composed of a certain amount of underlying spectra, right, underlying dye spectra, and somehow, of course, this can go to 30, 40, 50 of them, whatever, but somehow these will combine to form this observation that I saw. And we're trying to figure out how much it is. Note, again, I am not correcting these detectors for spillover or anything. The question I am fundamentally asking is, how do these spectra combine to form the observation that came out of all of the detectors? Okay? And so mathematically speaking, we're saying I have this observation. This is just sort of standard math. You have this is the sum over here. And I say that this spectra times an unknown amount of Pfitzy plus this spectra times an unknown amount of PE is going to give me this observation. And my job is to calculate these unknown amounts that actually underlied this observation over here, right? Again, multiply this by 40 different dyes, but in all my stuff, I'm going to show everything with just 1 or 2 dyes. And so how you do it, this stuff gets translated into usually matrix math. What is the matrix math for those who are not familiar? So this is the emission spectra of my dye. And what I do is if I just rotate it on its side and I put the numerical values of my emission spectrum, this is what goes into the so-called mixing matrix, or what was the spillover matrix. It's just the normalized emission spectrum of this dye. This is normalized so that everything sums to 1, right? So you replace your little picture with the numbers that underlie the value here, and you add all your dyes into it. You see this is my second dye. My second dye peaked in this detector over here. It's the highest value. Here, this first dye peaked over here. And you're saying, okay, well, if I take these 2 emission spectra and I multiply it by an unknown amount of Pfitzy and an unknown amount of PE, where this column represents Pfitzy and this column represents PE, I will get my observation also turned on its side. Here is the actual raw detector values that underlied my observation, or that was my observation. And some terminology that we use, this is a mixing matrix. Again, in compensation, when it was called a spillover matrix, but the more general terminology is a mixing matrix. These things that we're trying to recover, the amount of dye, are called the abundances. The things that we're trying to recover are the end members. This is used in many, many, many fields. This is not just in flow by any means. And so there's a very general term because things that people try and recover with this vary tremendously. So there's this sort of term that doesn't really mean anything called end members to represent what you're trying to recover. And this is my observation here. Okay? So this is transformed into matrix math, where the mixing matrix times my abundances, usually often represented by alpha, is equal to my observations. Right? And how is this often then taught? Well, this is a very simple equation. If m times a times alpha is my observation, I divide both sides by m. So I'll get that A equals O divided by M. And since in matrix notation you can't actually divide by a matrix, but you can multiply by an inverse, you get that A equals your observation times M to the -1, which is the same as dividing by M. And this was often referred to as a compensation matrix, M to the -1. And lo and behold, you get your A. And I think that if anybody has gone to any of my lectures previously or read any textbooks or read Mario's paper or anything, this is the way that this has been taught. I'm going to show this in a slightly different way. Okay, this is not a useful way of thinking of the problem as a simple linear algebra, simple division. Okay, so we're going to look at it from a more holistic approach and get back to this in the end, because this is what we're doing, but we're going to go a roundabout way that's a little more useful for understanding the problem. Okay? And the problem is dealing with the noise. Okay, so what we're really getting when we get an observation from a particular cell, we're not really getting the mixing matrix times alpha, which is the abundances of fluorescences that are on that particular cell. We're getting a mixing matrix times alpha plus some noise that's in the system, right? And the noise can come from various sources in various ways. I'm not going to deal with the details of that right now, but suffice it to say that we are dealing, the O that we have has all these components in them. Okay, and let's look at that and see what impact that has on us, right? So I'm going to do a simple one-dye experiment here with this very, very simple four-color mixing matrix. Okay, and you can see this mixing matrix peaks in the second detector. And I'm going to presume that we have 100 units of a particular dye on our cell. So if we multiply this mixing matrix by 100 units of dye, our pure observation should just look like this. I should have 30 units in detector 1, 40 units in detector 2, 20 units in detector 3, and 10 units in detector 4. Right? It's a very, very simple math. You just multiply each one of these by 100. So this was my pure observation if I lived in a noiseless world, right? But if I have a little bit of noise that's in my system that comes from electronic noise, background dark current noise from the detectors, photon noise, whatever you want to pretend the noise comes from, then you get that your final observation actually looked like this. So what you got out of your flow cytometer was the 30 pure signal and an extra 2, so you got 32. This signal dipped a little bit because of the noise, so you measured 37, even though the pure signal was 40, et cetera, et cetera, et cetera. So let's look at this for a moment, and we'll say, okay, the interesting thing about this is there is no value of alpha that can actually recover the O that you observed. There's no way to exactly calculate this. And let's look at this for a moment. If I know that 100 units of this dye, of this particular dye here, generated a signal of 30 in detector 1, but I actually saw 32, if I just look at this 32, I say, you know, there must have been more than 100 units, because 100 units would have generated 30 and I got 32. So there must be a little bit more than 100 units of dye been present there. But then when I go to the second detector and I say that there's 37, I say, 100 units should have generated a signal of 40, but I only got 37, so there must be less than 100 units of dye there. And here, there must have been more than 100 units of dye, because 100 units of dye would have generated 20 signal and I got 21. But then this detector is telling me there must have been less than 100 units of dye, because 100 units of dye would have generated 10, and I only got 9. So every single detector is telling me that there is a different amount of dye present on this particular cell. Right? It's a little bit crazy, right? And that's because of the noise. The noise is randomly screwing up this pure signal and making it a signal that there is literally no value of alpha that can perfectly recover your observation. And so what do? What we do is we actually take the question we change it to is not what is the value of alpha that recovered my observation. The question is, what is the most likely value of alpha that I had on this cell given this particular observation here? Okay? Because I know that I can't actually recover the true value, or in fact recover any value at all. All right? And so it turns out that the most likely value to have generated this observation was 98.3 for this particular cell. And if you actually multiply 98.3 times the mixing matrix, you actually find that even this most likely value does not recover your observation perfectly, right? There still is some discrepancy between your imputed observation or your calculated observation and the true observation. So even the observation that we recovered cannot fully explain this data. However, it is the most likely value to have been there given this particular observation and this particular mixing matrix over here. And in fact, it's really interesting that the actual abundance value of 100, which I literally simulated to generate this data, is less likely to have generated this observation, even though it actually really did generate the observation. But because of the noise, it's actually less likely that a value of 100 generated this observation than the value of 98.3, which is the most likely answer here. Okay, and I'm going to get back to that in a moment. So when we talk about most likely, we're talking about probability. And when we talk about probability, we need to discuss it in terms of a particular probability distribution or a particular noise model. And even if we are correct about the noise model and we assume the correct noise model, we will not necessarily recover the actual abundance that generated the signal. We are just going to recover a most likely abundance, which is actually very rarely the actual abundance that generated the actual observation that you saw. And you can actually calculate what is the likelihood here. I'm going to have to skip through this really quickly, unfortunately. But so if anybody recognizes this, this is actually just the normal distribution. This is the equation for the normal distribution given a particular mixing matrix and a particular observation. The point that this slide is that you can actually calculate an equation that given a mixing matrix and given an observation that you have, what is the most likely alpha and what is the most likely noise, or what is the likelihood of a particular alpha, a particular abundance, and a particular noise to have generated this observation from this mixing matrix given this noise model. And the point all I'm trying to do is point out that the noise model that you have is baked, that you are assuming is baked into the equations of what is the most of the likelihood. And then what you can do is you can actually do a whole bunch of math to find what is the most likely alpha to have given rise to this O given this M. Okay? And when you walk through the math, if I had more time, it's actually really straightforward how you get from a Gaussian distribution to this. It's not very complicated. It does take a bunch of steps that I don't have time to do. And it turns out that if you want to, if this is the likelihood of any alpha, if you want to calculate the most likely alpha, it turns out that for a Gaussian noise, this is the equation. Okay, so for Gaussian noise only, there is an analytical solution that will tell you, given this observation O, what is the most likely alpha to have generated this O. And when M is square, this reduces back to exactly what we're used to from compensation, that you multiply O by the compensation matrix to get alpha. Okay? So it is not simply that you are dividing your observation by M. The more robust way of thinking about it is that by doing this equation, you are calculating the most likely abundance value to have generated your O given an assumption of Gaussian noise, the OLS assumptions. Okay? And why is that more useful to think of it in terms of likelihood? Because it turns out that if you change your noise assumption and you change your noise model, then your likelihood function changes, right? This was what I showed you before, the likelihood of calculating alpha given this M and O for a Gaussian noise. But this is the likelihood of alpha with the same M and the same O given an assumption of Poisson noise. And of course, since these two equations are different, it is true, and it is that their maximum will be in different spots. So the most likely value for alpha given this assumption is different than the most likely value for alpha given that assumption. And so depending on the assumption of the noise model, you will unmix to a different value, is the point. And of course, you can complicate this as much as you like. Okay, so in summary, unmixing is the application of a mathematical model to your raw observations. Like with any modeling, the accuracy of your answers depends on how closely your model follows the true process that generated the data. When we unmix, we're calculating the most likely abundance value that generated the observation. Even when we do that, they are not guaranteed to match the actual abundance that generated the data, okay? Even the unmixed, they're not even guaranteed after unmixing to match the observation perfectly. You'll get a residual almost always. And even if you do match the observation, let's say you calculate an abundance value, you multiply that abundance value by the mixing matrix, you find out that that exactly matched your observation. Okay, so you think you recovered your observation perfectly. That still might not be the actual abundance that generated that cell's observation, right? There is a random element to it. And the key thing is maximum likelihood estimations require an assumption of the noise model. Even if your noise model is correct, all of the bullet points above apply. You are not necessarily going to recover the perfect answer on every cell. But for sure, if your noise model is incorrect, then you're going to be even further from the actual abundances that underlie your cells, right? So your model does have to match the data, but you don't think that you're somehow just doing a simple correction where you're recovering perfectly the abundance that generated that particular observation. All right, so that's sort of phase one of the talk. Phase two that I'm going to have to zip through much quicker than I wanted because I guess this isn't an hour talk, it's probably an hour and 20-minute talk. So is a little bit about dye collinearity. And I think people are familiar with this, is that you don't want to use dyes with two similar spectra, right? And here's a very simple example where the same cells were stained with multiple different panels, different two-color panels. And you can see here when the dyes are well separated, you have a very nice populations, they're tight. And as the dyes overlap, we get spread, right, in the data. The same populations are more spread out. So this has sort of been known. Why is this happening? I'm going to talk about why this is happening mathematically a little bit. The idea is to remember that the model is trying to find the most likely abundance values that gave rise to a particular observation, right? And let's do a little experiment where we put two dyes that were exactly the same into our panel. And this is the observation that we got from these two dyes, and we're trying to figure out how much of the two dyes there were. We're trying to figure out the most likely value. Well, this observation can be explained by there was all dye 1. It was no dye 2. This observation can also be explained by the fact it was all dye 2 and no dye 1. This observation could be explained that there were 50% dye 2 and 50% dye 1. And this observation could be explained even that there was twice as much dye 2 and a negative amount of dye 1. All of these combinations of dye 1 and dye 2 will perfectly explain this observation. And the fact is actually there's infinite possibilities of combining dye 1 and dye 2 when they're the same. And each one is equally likely as each other. And so there is no maximum likelihood. We have an infinite possibility of equally likely combinations of dye 1 and dye 2 that could explain this observation. And what happens is this results in a singular matrix, and a singular matrix cannot be inverted. And this applies even if two dyes out of your whole panel are exactly the same. Even if you have a 40-color panel, if two dyes are the same, the whole panel blows up. Okay? So this is obviously a very artificial example when two dyes are actually exactly the same, which never happens. But again, the point being that that's the idea, is the equations cannot find a maximum likelihood. And I'm going to skip these for lack of time. It's just explaining essentially, I'll just go to the end, that if I have two dyes, in this experiment I made two dyes. I made FitC and FitC that was shifted by 7 detectors, and I made FitC and FitC that was shifted by 1 detector. So they're not exactly the same dye, but they're very similar dyes. And long story short, what you find out is that when you unmix these with the same amount of noise, the FitC and the FitC Shift 7, they actually look, the CV is actually very similar to FitC alone. But if you add the same amount of noise to FitC and a dye that was shifted by FitC by 1, you see that the CV gets much broader. You know, this is a big increase in the CV between one and the other. Okay, given the exact same data, the exact same cells, the exact same noise model, the exact same abundance, just because the dyes were more collinear with each other, you actually see a numerically relevant, very easy to find increase in the CV of your unmixed values, because the model has many more ways to combine the shift 1 and the shift 2. And here's a nice illustration. What happened? Why is the CV worse when you have a shift 1? What I did is I found the cells that had the actual amount of FitC and FitC shift 1 on each of these cells was 1,000 on every single cell. So I found the one that unmixed the worst. And you can see here that what happened is in the FitC shift 1, there was one dye that unmixed that it had 678 units of FitC shift 1. That's quite a bit less than 1,000. Right, which is what was really there. And what happened is this signal had some noise in the peak. And in order to perfectly, or try as best as it can, the red is the pure observation that should have happened without any noise at all when the values were both 1,000 and 1,000. The blue signal is what actually happened. There was some noise. And what it can do, what the model can do, is bump up the amount of FitC and dramatically decrease the amount of FitC shift 1 and play with that so one can offset the other, and they have a lot of flexibility in having one balance out the other because the spectra are almost the same. And so it actually can have a large variance in these dyes to try and match the noisy observations. Whereas when the dyes are well separated, even though this dye, FitC shift 7, did come with a value 802 because you can see there was a noise spike that caused the observed value to be much less than what it should be. It couldn't really adjust this guy in any way to compensate or to offset this one because the peaks were so separated. So the model is much more constrained when the dyes are not collinear with each other. Okay? And that's basically what's happening. There's a lot of math you can throw at it, but the idea is that the more ways that the model has to combine the dyes together to explain the noise, the higher variance you're going to get in your unmixed values. And if the dyes are very separated, they're much more constrained in how they can mix together and offset each other to explain the noise, and you'll get a smaller distribution of unmixed values. And so people have come up with metrics for quantifying collinearity. It's been known for quite a while, of course, that using collinear dyes are not a great idea. And the issue is with all of these metrics is that they're pairwise metrics, okay? And they've always been done comparing the effect of one dye on another. I won't go into details about how they all work, because there's a problem with them all, okay? And the problem with them all is that dyes are not collinear just in pairs. Okay, dyes can be collinear with the combination of all the other dyes in a panel. Okay, and so we need a metric that compares all the dyes together at the same time. A pairwise comparison can give you a hint that these two dyes are not going to work together in a panel, but they certainly cannot quantify all the effects of the dyes interacting with each other. Fortunately, this type of math goes well beyond flow cytometry, and this is a problem that has been around well before flow cytometry got involved in doing math. And this problem dates back to as early as 1896, but was pretty much fully fleshed out in 1925. And the metric of choice for understanding this is known as the variance-covariance matrix. And for a Gaussian assumption, again, for an assumption of Gaussian noise, the equation for this is very simple, MTM, MT transpose M negative 1. And MTM to the negative 1 shows how the noise in every detector scales by virtue of the collinearity in the dyes, how that's going to unmix to a high variance abundance. And it answers the question, if I took this mixing matrix and I ran the exact same cell through this machine over and over and over and over again, and it was exactly the same, what would be the variance of the unmixed values that I get? In theory, you should say, oh, well, I should get the same value. It's exactly the same cell. But because of noise, you're not going to get the same value. Every cell, even though it was exactly the same, is going to have a different noise on it, and that noise will be manifested in the noise of the abundances. And this answers the question, what is the expected variance of every unmixed value dye? I'm going to skip this. So here's some, I'm going to show some examples. So if you have FITC alone, variance-covariance matrix is just one element, and you see the predicted variance is 4.95. If you have FITC and FITC shift 7, you see that actually it predicts that these two dyes basically are going to have the exact same variance as if they were by themselves. These two dyes are not interfering with each other at all. The diagonal elements are the predicted variance of each particular dye. But if you mix these two dyes together, you see now all of a sudden that the predicted variance increases quite dramatically. Okay? Now, these were all symmetrical, and you think, oh, are these always symmetrical? That's not the case. If the dyes have more complicated shapes, like here's a dye with 2 peaks mixed in with a dye with 1 peak, you can see that the predicted variances are asymmetrical. It doesn't always predict that every dye will have the same variance, of course. And here's a particularly interesting example, is that these pairwise metrics cannot capture the whole picture. Okay? So if you look at these two dyes, I have a 3-dye panel. If you look at the pairwise metrics of dye 1 versus dye 2, it predicts that the variance is 16 and 9 and has a cosine similarity if you calculate between these two of 0.7. If you take dye 2 and dye 3 and you look at their pairwise metrics, they actually have a very low predicted variance, incredibly low cosine similarity. And if you take these dye 1 and dye 3, again, slightly a higher cosine similarity, slightly higher metric. So if you looked at these pairwise distances together, dye 1 to dye 2, dye 2 to dye 3, dye 1 to dye 3, you would think that those were all fine. But if you mix them together in the same matrix, look at what happens to the VCOV. The VCOV predicts a destruction of the ability to resolve, a huge increase in the variance. Well, remember, the numbers before were 10 and 4. And if you mix them all together, it predicts a variance of 2,000 for dye 1, a variance of 717 for dye 2, and a variance of 780.77 for dye 3. And looking at the picture, you can see, because the combination of all of the dyes together are all collinear with each other, even though any pairwise pair is not that collinear with each other anymore. And this is what really matters, is the collinearity of all the dyes with respect to all of the other dyes in the panel. So pairwise metrics, certainly if you have two dyes that are collinear pairwise, they're going to be collinear in the big matrix. But you can get a lot of surprises where dyes that don't look so bad compared to any other individual dye don't work well in your panel together. Okay? So in summary, collinearity can be an important contributor to spread. And the collinearity, the reason why is because there are more possible ways to combine the component spectra together when the dyes are collinear to match your observation. And pairwise metrics, while they're helpful, cannot fully describe all of the interactions between the dyes in your panel. And the variance-covariance matrix or its related statistic, the variance inflation factor matrix, are the proper statistics to view all the interactions within the panel. And this is a little bit more about them. So the diagonal elements of the variance covariance, sometimes almost also called Kramer-Rao, who did some proofs on that, after some people who did some proofs on it in the 40s. The diagonals represent the predicted variance, and the off-diagonal elements can be used to diagnose which components, which interactions are helping to cause that variance. And so for the last, I might go over by 5 minutes, but for the last little bit, can we mitigate these effects of collinearity? Well, we can remove dyes that contribute to the high variance predicted by the VCOV, right? And how do we remove those dyes? Well, we can remove them from the panel. That's one way to do it, and replace them with other dyes. This is not always possible. When you have 30 dyes or so, some dyes will always be collinear to some degree or other with some of the other dyes. Maybe you can't find them experimentally. So what we did is remove them as what I will show in the next is a new approach that we just published is how to remove them as part of the unmixing process itself if you can't remove them from the panel. I do want to say removing them from the panel is the best way. I'm not going to pretend that it's not, but it might not always be possible. So this is a paper that we recently published. I think it came out just a few months ago in press. And so the idea is, what is an irrelevant dye? So an irrelevant dye is a dye that's present in your mixing matrix that's not actually on the cell itself. Okay? For example, that's not uncommon, right? If you think about a viability dye, you should not have any viability dye on any cell that you're actually analyzing because the viability dyes are excluded. Yet they're always present in the mixing matrix when you unmix your data, right? And if you look actually, we did an analysis and with all the caveats that how accurate it is, but basically from this OMIP 69 paper that's a 40 color dye, approximately the average is that there's about 16 dyes per cell, 16, 17 is what's typically there. There's no cells that had more than 28 dyes stained at the same time. So you can see that it, is intuitive to everybody and actually true that of course not every cell is staining with every single dye. So when you unmix with a mixing matrix that always contains every single dye, you have irrelevant dyes on the cell. So the question is, well, what's the big deal? If the mixing matrix worked, I should just get a value of 0 for those and they just sit around and they don't do anything. Unfortunately, it's not quite so simple, right? There is a very well-described understanding what happens if you include irrelevant variables in your model when you model, right? And given appropriate assumptions, you can accurately predict what including irrelevant variables in your model will do to the actual relevant variables in your model. This has worked out decades and decades and decades ago what happens when you do this. The irrelevant variables do not add any explanatory variables to the model, right? If I'm going to look here and I'm going to say, oh, well, if I go try to take all the people in the room and I'm going to try to make a model to predict how much salary they're earning and I put in their education level, that probably is part of it, right? But if I put the number of donut shops in the 3 square blocks from here as an explanatory variable for the number for your salaries in this room, that's not really going to explain anything, right? It doesn't have anything to do with it. So the question is, well, is that just doing nothing in your model, or is it actually bad to include the number of donut shops? Shouldn't it just come out that those have no effect? Well, it turns out that it actually does. Okay, and including irrelevant variables in your model can affect the variance of the populations of the relevant variables, right? And because what happens is those irrelevant variables can fluctuate to absorb some of the noise, and that means that your relevant variables are affected by your irrelevant variables, essentially. And that's due to the collinearity between the irrelevant dyes and the relevant ones. If you, in our terminology, if the irrelevant dyes that you're including have collinearity to the relevant ones, that when the model sees an observation, it can use those 2 dyes that are collinear with each other to balance each other out, even though one of those dyes is actually not even there. It's not even present. Right? If you think about if your viability dye is collinear with your CD3, it can use the abundance of the viability dye to balance out some noise in the CD3 dye, and it can give the viability dye some abundance, which will decrease the CD3 abundance, even though you know your viability dye is not even there, right? And that's basically what's happening in the modeling, all right? And this goes to show, again, to skip through, even if you just take beads and you add more and more and more and more dyes in the mixing matrix with the same beads, you'll get an increase in variance in your recovered beads that's very predictable by the variance-covariance matrix of that resulting mixing matrix that you use, okay? It's almost a perfect correlation between the variance that you unmix with and the variance predicted by the variance-covariance matrix, okay? And so the idea is to unmix the cell with only the dyes that are present on the cell. And how do you know which dyes are only present on the cell? Well, there's sort of two broad groups of ways to do it. One, you can use known co-expression patterns based on your a priori biological knowledge. You can use known staining patterns based on different subtypes. So if you know monocytes don't express A, B, C, D, and E, you could incorporate that into your unmixing by saying if this cell is a monocyte, remove A, B, C, D, and E from the mixing matrix dynamically as you're doing your unmixing. We chose the following approach that doesn't require a specific biological knowledge about the sample. And what we did is we unmixed the unstained control with all of the dyes in the mixing matrix. And then we calculated the 99.5th percentile of the distribution of each unmixed dye. And when unmixing every cell in the sample, if the unmixed value for that particular dye was below the cutoff, we presumed that dye wasn't really there, and we removed it from the mixing matrix. And we kept looping and looping and looping until all of the dyes were above this cutoff that was determined by the unstained control. Long story short. And I guess I'm out of time, so I will just come to the punchline here. This is going to show that we included 2 dyes in the mixing matrix that did not exist at all in the panel. So this is when we use only the dyes in the panel. This is when we added 2 dyes that didn't exist in the panel. And the true OLS, so this is just OLS unmixing in this corner, the true OLS had detected correctly that 99.6% of the cells, didn't do it perfectly, there's a few percent that got away, but 99.6% of the cells actually had a value of 0 for Pacific Blue and SB436, which were not actually there. These were artificially added to the mixing matrix. And in fact, when you unmix this data with true OLS, it looked exactly like the 2 dyes weren't there, which is not surprising because on 99.6% of the cells, it actually removed those 2 dyes from the mixing matrix, so it was back to as if those dyes didn't exist, and you get back to the same picture. Okay? Similarly, we actually added those bad dyes to the panel, and the interesting part of this one is that this is the unmixing via OLS. And if you gate on these cells and you look at what these cells looked like under true OLS, you find that most of them were in the same sort of similar region, but there were a very few number of cells, about 1.5%, that were clearly TCR gamma delta negative, even though they showed up in the TCR gamma delta positive when you gated them by OLS. And when we did some further investigation in OLS, we found that these cells exclusively came from this population over here that was assigned this completely non-biological value for BUV395 of negative 10,000. Okay, so for whatever reason, the unmixing, people tend to gloss over the fact that their negative cells go from like negative 10,000 to positive 10,000. They squish it on the bi-exponential. But in fact, because these cells were so negative for BUV395, they got a artificially high positive for BV421 to counterbalance them. The 2 dyes are collinear, so it took 1 dye hugely negative, 1 dye hugely positive in order to match the observation. And in fact, when you look at them, you find that even though the value is different, it matches the observation almost as well It is slightly better to have these non-physiological values. Very, very slightly, it matches the observation. So for OLS, this is the maximum likelihood because it matches the observation ever so slightly better than if you remove it, but you can see the difference is minuscule. And this is, of course, a much more biologically relevant value. There's no real reason why this was negative 10,000. That's just because of the noise in the instrument, right? This is a similar type of analysis where we showed that the true OLS approach gives you what is arguably biologically more relevant and interesting results by removing dyes that are causing collinearity. And this was another sort of last thing where the problem that you get when you do true OLS is a lot of your data is right at zero, because it removes a bunch of dyes from certain cells and assigns them to a value of exactly zero. These are zero for one of the dyes, these are zero for the other. You can't even see the little dot that's there that is the zero that is double negative. And as biologists, we actually rely on this artificial blur over here that's completely fake in order to do our gating on the negative population. And if there's one tiny little dot here, you kind of don't see it, even though it might be 95% of your data. So there was sort of a negative consequence, although it was more biologically accurate to say those cells literally had zero abundance, particularly for an unstained sample. What's the point of getting a giant blur? You literally know there's no CD4 because you didn't put CD4 in there. So there should be a little dot at zero because there's no anything. And so in every channel, there should be on your dot plots of your unstained control, there should be a little dot at 0, 0 in every dot plot. But of course, we don't get that, right? We get a blur around 0 because of the noise and the variance and the covariance in the mixing matrix. But that is not useful for gating. So we sort of came up with a strategy saying, well, if this is totally artificial, and it is, the distribution around 0 is completely artificial and only a result of the noise in your system and the peculiarities of your mixing matrix, we're going to artificially make that negative data match the unstained control. All right, the unmixed values of the unstained control to give you something to gate on. All right, and that's another sort of thing. And arguably, I feel like this is a more reasonable way of demonstrating the negatives versus the positives in almost all these examples than this artificial blur that's equally as artificial. And that's it. So that's all I wanted to talk about, really. A couple last summary slides, but I did want to acknowledge my colleague, Ryan, who works with me, and all of these people here provided really extensive comments on the manuscript and on the work we were doing. And so I'll know to cut this down next time a little bit more. So thank you very much.
So any questions? We'll take 1, 2.
Not so much a question as a comment, or a very, very big thank you, actually. First of all, for shedding light onto the behind the scenes. How is compensation and mixing actually handled? I think for a lot of people, you know, you just click a button and hope for the best, maybe, you know, look at the matrix and fiddle around. But to actually, you know, see how it's done, I think that's very instructive. Last year, OMIP 102 already experimentally demonstrated the issue of having irrelevant dyes in the matrix and how that can mess up or just blur the resulting data. So it's really beautiful to see this explained on a mathematical level. And then, of course, you know, you being you, finding a solution to it as well and kind of improving the outcomes. And we all know that, you know, even with the best ever compensation controls, we don't always get a perfect matrix. And people go, well, you know, maybe it wasn't the perfect control after all. Was there this? Was there that? Did I pipette wrong? Whatever. And so, you know, to kind of hear you explain it so crystal clear that it is a problem of the math behind it doesn't mean that, you know, things in the lab don't go wrong. But, you know, there's a level to it that we don't control as, you know, as experimenters, as people holding the pipette or, you know, working on the cytometer. I think that's put some minds at rest and is put some hope into all of us that we might actually get a perfect resolution of all our dyes and all our cells, etc. So thanks so much, David.
Thank you. Yeah, and the issue is again, this is all presuming, this is even if your spectra for your single stain controls was correct, you're going to get these problems, these collinear. If they're not correct, for whatever reasons, and sometimes you can't do anything about it, and we don't even understand why sometimes they're not correct, it just gets worse, right? Your model is wrong because your model is saying I'm using these spectra, but your data was for whatever reason using another spectra, and that will further increase the variance and the spread in your unmixed values, unfortunately.
I have one quick question. I know we're way over time, but this makes sense mathematically, Dave. It's beautiful. But biologically speaking, right, with monocytes, you said, you know, you can remove viability because they're viable, but monocytes or other cells that tend to sample the environment will always have a degree of background with that viability because they've taken it up, right? Mm-hmm. And then you also have the case, like Sasha mentioned, with trogocytosis, where you have T cells that could potentially nibble on a cell surface and pull it, such as like with CAR T cells with CD19, they might, you know, have CD19 expression on them. But if you go in with the mindset that, oh, you know, for CAR T cells, we're looking at them CD3, they should not have any CD19. But what happens in the case where you may have that instance of trogocytosis and you do have CD19 expression on the T cell because of that nibbling? How would that impact the unmixing?
Yeah, I mean, it's not, there's no perfect solution, right? So it's kind of like if you include this other dye, and this other dye is collinear to other dyes, and this other dye is only present on 0.1% of your cells or present to a low degree, it'll mess up every other cell in your unmixing, but may be arguably a little bit more accurate on that 1% of the cells where it's actually there. Whereas the corollary is if you remove that dye completely and incorrectly, then you'll get better unmixing on the 99% of the cells where it actually wasn't there, but you might mess up the 1% that is there. So like with anything, you have to know what you're doing with your system. Hopefully, if it was taking up some of this dye, then it would be taking it up in excess of the unstained control, and it wouldn't be removed from your sample because it actually is above the amount of fluorescence. It's hard to argue that it's taking up some of this dye in a significant amount, yet it didn't increase its fluorescence expression above and beyond the completely unstained control, yet it's actually still there, and it's significant, but it's so dim that it was within the unstained control distribution. So, you know, but I'm not advocating that way that I'm describing is the be-all and end-all. There's any way that one can think of that makes sense for their experiment to remove particular end members from interfering with others will work, and not every way is appropriate for every single sample.